also has quite a bit of protozoa and other things. So lawn manure is just lovely stuff. Strange, you know, for manure is lovely stuff.
I agree.
Relating to her question, I've heard that animals that have a lot of antibiotic and hormone injections to the manure.
Yeah. You have to look out for horse manure, especially if you're getting it from a stable, because they're going to be putting deworming and that can really shut everything in a compost pile. Now, it is a sterilant. So think what you're doing to your horse's digestive system when you use a strong sugar. You're wiping out the biology in your horse's digestive system. So maybe we ought to be giving our horses some compost tea. After the deworming medicine has done its thing, then toss down some compost tea and reestablish the right sets of organisms in your horse's digestive system.
Whatever that is.
Well, it's a good, the forestomach on the horse is aerobic, but then the back stomach is anaerobic. So they need both.
So I have a neighbor who's a vet, and they're constantly cleaning out their, you know, it's sawdust and horse manure and all that. So is there anything that, I mean, what would you do to sort of remediate to that worm medicine?
You can't put that much of that manure into the compost pile. So you no more than 5% or 10% of that manure that's got the deworming medicine in it should go into your pile. Otherwise, it really does shut down the organisms.
But if it was like, is there like a half-life of that medicine or something? Do you know what I mean? Like if you just left it stand for a month or 6 or whatever, could you?
I don't think there are any really good studies on that. I haven't really looked into that, so you might go in Google strongylid decomposition rates or something like that to figure out what the half-life of that material is. And we sometimes get horse manure from people who say, "Oh no, I've never used a deworming medicine, never done it." And we get their horse manure, and it's like, "Yes, you have, sweetie," because we are absolutely not getting temperature in this pile. And then they go out and ask their vet, and their vet says, "Oh yeah, I've been doing it, you know, every 2 weeks." Thanks. People don't know what their vets are doing sometimes.
Is there a certain interval after the deworming treatment?
About a week.
After that, then it's okay.
Yeah, and it's okay. So, yeah, and the problem is like in public stables, they feed these 10 horses deworming medicine, and then the next 10, and the next 10,
And then it's hard to keep track.
yeah. Did you separate that manure from the bad manure from the good manure? It's sometimes just not worth it to deal with horses because yeah. Now the antibiotics, most pelletized plant food material for cows and horses and pigs and sheep and Purina cow chow. It's got antibiotic in it. Don't worry about it. Antibiotic is a protein. There is somebody in your compost pile that's going to regard that antibiotic as yummy, yummy, hearty food. So it's going to get taken care of. I don't worry about antibiotics at all.
Cows. Is that done?
About cows?
The deworming for cow and sheep and goats.
Yeah, I would probably not want to use that manure. So deworming medicine in general, I don't want to use that manure in my compost pile.
It reminded me of a story. I have a friend that lives near Tracy in California, and they have a feedlot not far from where it is, and they have huge piles of this stuff. And I was thinking, whoa, there's a reason for it. That's why they can't get rid of it.
Yep, no one in their right mind would take that stuff. Because you're going to turn your soil into a toxic waste dump. Yeah.
I'm sorry, I think on a loop here. What was the reason? Deworming medicine.
Deworming medicine, nothing, the fungus doesn't grow, right?
I see. Okay.
Yeah, you pretty much shut down microbial decomposition. That strongyl material is really nasty stuff. It's a steroid. So strongyl, methylbromide, same category.
But you said just for wheat, right?
Yeah, so when they feed the animal the deworming medicine, what comes out for the rest of that week is really, don't use it. But then after that, so if they're deworming their animals typically once a month, you know, for that 1 week, you wouldn't want to use that material. If they go a little bit longer before they deworm again, especially in private stables, they'll tend to deworm all the animals. And as long as they're not seeing any worms in the manure, they won't do it again until they see a problem start to develop. Ivermectin, not a problem. It's an antibiotic, so it's very rapidly decomposed by quite a number of microorganisms.
Would you
It's
use,
not as nasty.
sorry, did you use dog poop?
Sure.
Just like chicken poop?
Yeah, it's manure. As long as you make sure that that poop gets the hot treatment that it requires, you're going to kill all those bad things that might be in there. And with cat manure, dog manure, the real problem is the worms. So you've got to get it through the heating process. So heat a little bit, make really sure, well, especially like with a pile like this. Notice that, you know, some of the stuff is kind of escaping out the edges. You're going to have to make sure that all of that stuff gets into the middle of the pile. You know, 6 weeks from now, when that's a finished pile, don't be taking some of the from over here and added it back into the pile. Every part of that pile must get up to temperature, hot enough, long enough. So don't be contaminating your pile by putting something else in there.
And now, I've heard a question that a way that could be remediated would be feeding a compost to composting worms.
Yeah, but why go through a process of hot composting and then put it into the worms? One of the things that people will often do is put a compost pile together like this, or even straight manure, and get all of that pile up to really high temperature for 3 days. And now, as long as it's up to high temperature, you've dealt with the worms and the it's really weed seeds that you have to worry about with worm composting. So get it up to high temperature so you've killed the weed seeds. 3 days. So now spread that material in real shallow layers so the heat that might be generated isn't going to kill your worms. And so now all the rest of the pathogens are dealt with by somebody else who turns your pile for you. See, the lazy gardener's way of making compost. So heat it up nice and hot, and then take that material that's been hot for 3 days, that goes into your compost pile. And then let the worms turn and stuff for you.
I've heard that the pathogens, human pathogens, don't survive in a worm gut.
That's right. So when we talk about compost day, I'll go through that. Okay.
I'm sorry. Jerry, repeat again. I'll think that up because I was.
Let me write it down. Now, this will be in your notes from the compost day, but when we think about that pile, it's the center part of the pile that's going to get hot. And so, how much manure do we have in there? You know, is pretty much all of the center going to get up and hot? Is it just a small part? That's where we want to be sticking our thermometers into the pile and figuring out, you know, do we have a high enough temperature through most of the pile or just a small part of the pile? And then we want to get everything in that pile into that hot center at some time. So we'll stick our thermometer in tomorrow morning. You cannot consider that you're really actually composting until your temperatures in the center of the pile get above 131 degrees Fahrenheit. If you're only up to 131 to 150 in that range, you're going to have to let that pile heat at that temperature for 3 days. And it has to be continuously above 131. If it drops below 131 for even a few minutes, don't let your certifier catch you doing that. So it's got to be above, or the E. coli starts to resuscitate, and then you got to keep it there for another 3 days. But if we can get it up to 150, somewhere between 150, 160, we probably only have to leave it at that temperature for 2 days. Higher the temperature, the less time we have to keep it hot. If we get up around 160, then we really only have to leave it there for about 24 hours. So when do you have to turn this pile? How long has it been at one temperature? So we walk in here tomorrow morning and it's already at 160. Oh, we only have to leave it at that temperature. So we could come in Thursday and turn the pile so we get the outside material into that hot inner core. And then watch it heat up again. So what temperature do you reach? Now, a lot of this information is based on the work that has been done in food microbiology. So for 100 years, 150 years, we've known this kind of information. It has not percolated through to the soils people. So they're stuck on, it's got to be above 131 for a full 3 days, and you can't turn until you're, no matter what temperature you actually get up to. So we have a little education that needs to happen with soils people. Because in food microbiology, this has been done to death about what the temperature is and how long you have to hold it to kill off any of the human pathogens. So, a little bit of fun there.
What happens if you see ash or gray material in the center? What does that tell you?
That ashy material, so sometimes as you start digging into your pile, you'll see just below the surface a layer of white ashy material. This means you're starting to go anaerobic. So in cases where people grind all of the material in this pile too fine, so it's just like you've got a pile of clay, and because we have not built good structure in that pile yet, bacteria and fungi haven't really gotten going yet. We don't have the good structure. So when you see that layer of white ashy or gray ashy material, what you're actually looking at is actinobacteria. And that is telling you, because the only time those actinobacteria are going to start growing in that pattern is you're dropping into anaerobic conditions. So you see a layer of actinobacteria, turn your pile right now. The temperature in the middle may not say turn, but this is telling you you are going anaerobic and you do not want to have high levels of actinobacteria in your pile unless, oh darn, there's an exception. So more for you to think about. Actinobacteria are really important for non-mycorrhizal plants. So if we're dealing with the mustards, we're dealing with brassica, kale crops, broccoli, cauliflower, these are all plants that should not become mycorrhizal. Now, other plants can force mycorrhizal colonization on these guys, but then the other plant is sucking the living daylights out of your non-mycorrhizal plant. There's some really interesting work by the Allens, Mike and Edie Allen, down at the University of California in San Diego. And they have several experiments where they're looking at like sagebrush. Strongly mycorrhizal plant, it has to be mycorrhizal. So in that root system down in the ground, very mycorrhizal. Good colonization, and the hyphae are off into the soil, gathering nutrients for your plant. If that sage is growing close enough to, like, say, a field of broccoli, and I believe the work that they did was with broccoli. And she spelled, no, there's only one element, broccoli, close enough. What they showed was the broccoli was growing just fine, and then it became colonized with the mycorrhizal fungi from the sage. And the broccoli started to wilt. It started to do very poorly. So they had a person that came out and labeled the broccoli with N15, labeled nitrogen compounds. So injected that into the stem of the broccoli, and they documented that those proteins, that good food resource, moved from the broccoli into the mycorrhizal fungus and into the sage. Broccoli died. Sage did very well, thank you. So while you can sometimes find colonization on these brassica, it is not beneficial to them. So how does the broccoli protect itself from this kind of interaction? It puts out a whole bunch of exudates that feed the actinobacteria. And the mycorrhizal fungi coming on over here going, oh, look at this cute little broccoli. Ah, it's got a protective shield. Oh, no. So those actinobacteria are protecting that root system of your brassicas and your colon kale crops from colonization by the mycorrhizal fungus. So if you're growing these plants, seeing those actinobacteria in your pile is, well, it might actually be exactly what you want. Now, any plant that is mycorrhizal, do not put that material anywhere close to a plant that is mycorrhizal, because actinobacteria, or at least in most of the cases that we've been looking at, actinobacteria suppress mycorrhizal colonization. Not all of them, so there's a few exceptions, but really for mycorrhizal plants, if you see that actinobacteria layer in your compost pile, turn it. Turn it right now. If this compost is going to be going on brassicas, then, well, maybe don't worry about it so much. So, you know, people always ask me, what is the right set of biology to have in your compost pile? Well, it depends. Because what are you trying to grow? What's the biology that we need? So when we look at succession, trying to understand this interaction between these microorganisms in your soil and what's going on during succession, where do these different plants fall? So let's go through this concept of succession and then how we discovered what's going on with bacteria and fungi, protozoa nematodes in the soil. Is there a question?
I wanted to ask, I read some literature that suggests putting soil into your compost. Is that kind of acting as an inoculant, or what do you beneficial thing?
I only put additional soil in is if it's a really good fungal soil or it's got really good protozoa, and put that in as an inoculum. So soil, if you're just putting soil in for the joy of putting soil in, nah, don't bother. You're just making your compost heavy and difficult to turn. Maybe if you're a commercial salesperson and you want to have more weight of compost so you can make more money, okay, that works, but then it's not really kosher. You're paying compost prices for dirt. Not really. Now the Lukis, which are some of the people who carried on a lot of composting trials in Austria, and they offer a composting class that's really kind of fun to go take, as long as you can speak Austrian. So they always recommend to put 15% clay into the compost. And they claim that by putting that clay into the compost, you're putting more surface area for your microorganisms to grow. Well, look at all the surface area and all that organic matter we just put into the pile. Are we lacking surface area in there? No. Where's the food resource in the clay? It's mineral material. Do you need any more minerals in that pile? No, because think of all the nutrients that are in that organic matter. So I just don't buy it that you need to put clay into the compost. Well, I put clay into my compost and it composts lots better. That's telling me that you have a whole bunch of toxic stuff in that organic matter and the clay's absorbing it. Do we have to put clay into your compost pile to absorb the nasty salts and the toxic materials that might be on some of your organic matter? Put some humic acid in there if you're concerned about it, because that's way more effective than the clay at absorbing those kinds of materials. So clay, putting soil in there, yeah.
What about doing it on cement or wood?
On cement, it's easier to know exactly what actually goes in the pile. So sometimes putting out a cement pad, a little bit easier to know really what's in your pile. Because like tomorrow when we turn this, how far down is our pile really? Where does the pile end and the soil begin? Sometimes it's hard to see that demarcation. So come the time when we're finished with this pile, we've got to make certain that we don't put any non-composted material in with the composted material. Not composted material could still be chock full of pathogens, weed seeds, problem organisms. So we don't want to contaminate our pile. Hard to tell sometimes when you're in an area like that. After we've composted 10, 20, 30 times, and everything in there's been composted, so we don't have to worry about it. A lot of times when you talk to municipalities, when you go get a license to allow you to make and sell compost, when you go from making it for yourself to you are now going to sell it, the legal system will require that you have a composting pad. Because so many people have put piles of compost together, allow it to become completely waterlogged, and then they leach out all these horrible pathogens, all that it is, draining off and running into the local stream or into the ground and into the groundwater, just chock full of pathogens. So because we've had people that have inappropriately composted, they have started to write regulations that you have to compost on a tarmac pad or a concrete pad or something where all of the leachate coming off goes into a pond, and then you've got to treat that water so it doesn't have any human pathogens in it. So as you go into commercial composting, it becomes more difficult because you've got to make sure that you're doing a good job. If you're selling people something chock full of pathogens, if you're destroying water quality around your composting operation, that is.
So that operation in Australia that was doing the composting on their own property with their own cow manure stuff, using it themselves is fine, but then if they start to have too much and start to sell it as a secondary product, that's when those regulations will come in.
That's typically, now sometimes in some states, it's if you produce over a certain amount that you sell to people. So sometimes there's a volume. If you're only selling 20 or 30 tons of compost, state doesn't care. You start selling tons and tons and tons, that's when the rates kick in.
These compost facilities in Canada that don't have
So the regs here in Hawaii don't require the concrete pads?
I don't know, I think so large operation in Canada, they didn't have any concrete pads.
Just wait. Or if that composting operation is careful about their compost and they never screw up, we might never get those regs in Hawaii, which would be really nice. So, succession. Let's go into an understanding of how biology changes as we go through succession. Now, I couldn't resist pulling this picture out of this book. This is a recent textbook, ecology textbook, and shows very clearly, we start off with, bare soil, and then the weedy system, and then grass comes in, and then here comes the shrubs, and now we get some trees, early successional trees, and then old growth. So great, as we go through this plant successional system, the birds that you find in the system go through their own successional process. Mammals in the system go through a succession. Insects in the system change as that plant material changes. But please notice, what did they forget in this picture?
Weeds.
How about soil? Does succession go on in the soil? What percent of the system are they actually looking at above ground versus what's going on below ground? Total ignorance. Complete ignoring of probably the thing that controls that successional process. So we get that soil biology in there. We're going to change so many of these problems. We're going to stop erosion, stop runoff, all of these problems. We're going to stop the weeds from growing. We don't have drought problems. We can fix compaction. We aren't going to lose our nutrients into water, so we maintain water quality. get the carbon back into the soil, all of these good things. So understanding this food web and how it changes as we change the plant in the system. I want to look at those sorts of things. Just to review the critters in the food web. Let's go through this one. Sunlight, carbon dioxide, photosynthesis, all those sugars go down into the root system. Dead plant material from above ground, dead root material going into that organic matter box. Is it more bacterial food or is it more fungal food? So we're feeding bacteria the simple, easy to use sugars, not complex, not difficult to have a good time partying on that stuff. Fungi, they use more complex food sources. They're going to sequester a lot more carbon. When we think about bacteria and the way they grow versus fungi and how a fungus grows, if we compare the same simple sugar, so we have a 6-carbon sugar, for example, when we look at what bacteria do with that carbon in that sugar chain versus what fungi do with the carbon in that sugar chain, bacteria taking that sugar, they're going to blow off 60% of the food that they use as carbon dioxide. So they start chewing on a sugar, most of that carbon off back into the atmosphere as CO2. 20% of those carbons are going to be used to grow new bacteria, so it goes into reproduction. And 20% of that carbon is going to be released as a waste product that now some other microorganism, bacteria, fungi, somebody, is going to be using that waste material. So bacteria, when they grow, are not conserving at all of the carbon that they utilize. When we look at fungi, same sugar, if they can use it, only 20% of that sugar molecule is going to be lost as CO2. We're going to grow 60% of that carbon is going to grow new fungi. And 20% is going to be released as waste material that some other microorganism is going to be able to utilize. So if we're trying to put the CO2 in our atmosphere back into the soil, do you want to be growing bacteria or do you want to be growing fungi? Pretty simple. In general, on average, fungi are going to sequester a lot more carbon, a lot more of the nutrients in their biomass. The question might come to your mind, well, so why does it work that way? Well, when we're looking at bacteria, their carbon and nitrogen ratio is very narrow. CN ratio on a fungus is going to be highly variable as you look at the age of that fungus. So we're going to go through that discussion probably tomorrow. So fungi competing with bacteria, they very rarely use the same food sources. So fungi on more recalcitrant materials. Recalcitrant, isn't that a fun word? What am I talking about when I say recalcitrant? Do you have recalcitrant children? No! My grandson's favorite word. No. Want to go to bed? No. Want to stay up? No. Want to have ice cream? No. So, recalcitrant. Everything is gone. So when we talk about carbon, what do we mean by a recalcitrant? Well, when we start looking at a simple carbon chain, this is a simple food resource. No branches, really easy. Well, bacteria are going to win on this. And a competition between a bacterium and a fungus for this simple sugar, bacteria are going to win. Bacteria make the best enzymes on this planet for grabbing any kind of carbon. That is the bacteria. As long as it's a simple chain like this. So we can get carbon chains that are 100 carbons long, 1,000 carbons long, so you can get really long, simple carbohydrates, and it's going to be bacterial food. But as soon as we put a branch Now we've just reduced the number of bacteria that can utilize this material. And now a lot more fungi are going to win in competition with bacteria for this food source because of the branch. Fungi make multiple enzymes that can deal with any of the linkages. Bacteria can only make enzymes that chew on the ends. So anytime we have this kind of configuration, bacteria are going to have to regear their enzymatic metabolism in order to chew on these carbons. Fungi don't have to retool. They're already making those enzymes. So imagine now if we, a couple thousand carbons going that way, now what if we put a chain here? What if we put a branch over here? Now we've got carbons going this way, maybe throw a sulfur in there, who knows? Add a few other materials in there. But the more we branch that carbon chain, the more this material becomes recalcitrant. Only fungi can break it down. Bacteria can't touch it. So we start to get more and more complex. So if we put another 10,000 carbons, each with branches, and all of these branches have branches. And I'll go that way and keep going. We'll go this way, we'll go that way, we'll go that way, and I'll just make this a big spider molecule. Each of the ends, that end over here can bind with this end over here, and that end binds with that end, and that binds with this, and then see how it becomes kind of like a piece of chewing gum that somebody kind of got it all nice and gnarly. Extremely fungal food. There is not a bacteria on the planet that can touch it. Humic acid, that's the structure of humic acid. Extremely complex, lots of branching, bacteria can't even begin to deal with it. So lots of interesting differences between what bacteria can use and what fungi can use. Now continuing on in our food web, nematodes, so root-feeding nematodes. Okay, they are secondary trophic level, so here's our primary producers. Secondary trophic level or our primary consumers. Nematodes eating the root system. Bacteria then are eaten by protozoa, flagellates, amebiciliates. They can be eaten by bacterial feeding nematodes. Fungi then are eaten by fungal feeding nematodes, or they can be eaten by fungal feeding microarthropods. Root-feeding nematodes can be eaten by those arthropods too. Well, root-feeding nematodes are also eaten by the predatory nematodes. Here we go. Look at it. There's a missing arrow in our diagram. USDA did not put that arrow in there because it made the picture too messy. Truth versus messy. Who won? So this is originally my drawing on the food web, and they just left a few arrows out because they thought it was too messy. This is published in the Soil Biology Primer. That is available online if you go to the USDA website. That Soil Biology Primer is online, and you can use any of the pictures and any information in there that you want. It is public domain. Let's see, out of the, I think there's like 9 chapters in this book. So out of the 9 chapters, I wrote 7 of them. So I think it's a great book. I think it's absolutely wonderful. What's the name? It's called the Soil Biology Primer. And it was published by the USDA.
And you said we can get that out of their website?
Yep. Yeah, you should be able to find it on their website. USDA for the Soil Biology Primer. The last time I looked, it was online. But every once in a while, the USDA makes things disappear, and you can't find them anymore. So there should be another arrow from the root-feeding nematodes to the predatory nematodes, because this is a great little organism to wipe out your root-feeding nematodes. They really love root-feeding nematodes. So now, think about you've got the arthropods eating the fungi, nematodes, fungi and bacteria, protozoa eating your bacteria. Do we need to have these higher level predators in the system? Do we have to have things that eat this level of nematodes or microarthropods that eat other microarthropods? How critical are they? Well, let's say you had nothing to control your protozoa, your nematodes, your microarthropods in this stage. You could get so many of these organisms growing that they would overeat their food resource. And then they shut down nutrient cycling and they stop building soil structure. We don't have any protection on the root systems of our plants because the predators can overeat their prey. So these guys are critical to have in our system because they keep these guys in balance so that these guys can continue to do their job. So that's critically important. What if we get too many of these guys? The same thing. If we get too many of them, they overeat these guys, and all of our nutrients end up in a bacteria and fungi, nothing available for your plant. So we gotta have the higher level predators. Notice that earthworms aren't really clearly put into this picture. Most people think of earthworms as being decomposers. Earthworms eat organic matter. No, they don't. Earthworms do not make the enzymes to decompose plant material. Sorry, they do not belong here. Earthworms are higher level predators because they eat anything that they can get in their mouth and crush to death. So earthworms, higher level predators. We gotta have those guys because we gotta maintain balance at every step along the way. So who eats groundhogs? And who eats scurries? So above ground, we've got more steps in this food web. When you really think about how many steps there are in the food web below ground, now here comes our carbon source, and so our bacteria are going to eat it. And then flagellate eats your bacterium, and then amoeba eats the flagellate, and then a cilia eats the amoeba. So we're already at 5 trophic steps here. And then that cilia is eaten by bacteriophagic nematode. So now here we are at 6. And then nematode, a predatory nematode, eats that bacteriophagic nematode. We're at 7. And then a microarthropod eats that predatory nematodes. Here we are at 8, 9. And that's when we get above ground. So when someone wants to talk to you about food webs all can only have 3 steps. Above ground, there's this attitude that we only have 3 steps in the food chain. So we have grass. Grass is eaten by a deer, and the deer is eaten by the mountain lion. 3 steps in our food chain. Does not happen below ground. This is a food web and way more stable in a system like this. So that the fact that this and that and the other thing all can eat each other makes that below ground system extremely stable as you develop complexity in the system. Question?
Yeah, with the earthworms, I mean, we're feeding them like table scraps.
But the earthworm just chews up the organic matter. It can't consume that organic matter. So when we look at an earthworm, so we'll throw this one in here now. I am so distractible. Here's our earthworm. And it's going to take a bite with its mouth of your mango. But it doesn't have the enzymes to actually break open the cells of the mango. It cannot decompose that organic matter. So what it's actually looking for are all the bacteria and fungi, the protozoa, the nematodes, that are growing on that organic matter. It's the bacteria and fungi decomposing your mango. Protozoa and nematodes eating the bacteria and fungi. So takes a bite and immediately pushes that organic matter into the next organ in its body. And this is just like a crop on a bird, a gizzard. And so this muscular organ crushes that mango material. And any of the bacteria and fungi that are caught between the surface of that muscle and the mango, any rocks, any pebbles, any mineral material in here, may cause those bacteria, fungi, protozoa, nematodes to pop open. And it releases the juice from inside that animal. And what the earthworm actually takes up is the juice from those organisms that have been crushed. As the earthworm takes another mouthful, that little bite of mango moves in a little bit further, crushed again. More organisms pop open, more food for your earthworm. Moves on, crushed again. As it moves into the esophagus in the earthworm, again, it's just a crushing organ. And so that mango moves in there, crushed. Crushing out some of the bacteria, fungi, protozoa, nematodes. Moves a little bit further, moves a little bit further, and every step along the way being crushed down, and the organisms on that material releasing their cytoplasm. As it then moves into the rear end of the earthworm, this is a culture chamber. Some of the absolutely best bacteria, fungi, protozoa, and nematodes that are found in soil live in the rear end of the earthworm. So it's a culture chamber. And so as that mango, whatever hasn't been crushed or decomposed up to this time, is now going to be utilized by the bacteria and fungi growing in this chamber. And we're going to grow billions and billions of bacteria, miles and miles of fungal material, protozoa, nematodes. Man, there is nutrient cycle going on inside this earthworm. And as one organism eats another, we're releasing cytoplasm, and that's what your earthworm is actually taken up. So now that little worm poop, otherwise known as cast, worm cast, contains an inoculum of some of the best microorganisms that you want to talk about in soil. Earthworms, strict aerobes. If you go anaerobic in your materials, the earthworm's leaving. They cannot tolerate anaerobic conditions. So like when you've got a worm bin, and somebody says, well, in order to keep your worms in your worm bin, you need to string lights to prevent your earthworms from leaving. Why would they want to leave your earthworm bin? You're feeding them every day, aren't they? Aren't you? No, why would they ever want to leave? Because things have gotten so bad. It's gone so anaerobic in your worm bin. And so they're going to try to take off. And if you strung lights across the top, the information is not going to come through to you that things are really bad in your worm bin and they're dying. It's going anaerobic. It's nasty in here. Wouldn't you like to know that things are going south and all as your earthworms try to crawl out of your worm bin? No lights. Don't put your earthworms in prison. So they are higher level predators. They do not actually eat that organic matter. It is the microorganisms inside the earthworm that eat it. And then the earthworm eats the organisms.
So all they do is chop it up so the other guys can get it.
Yep. So more growth of bacteria and fungi because now there's more surfaces, there's more edges. So the earthworm is just a conduit. Yeah. Okay, so we talked briefly about this before, but this is the work that we did that first started to clue us into the fact that as we go from very early successional systems that have a lot of weeds, highly disturbed, strictly bacterial, a little bit of fungi, a little bit more fungi, quite a bit more fungi, fungal dominated. First time ever that this was demonstrated. And so it clues us into the fact that as we want to increase productivity in an ecosystem, we have to change that ratio of fungi to bacteria, protozoa, and nematodes. As we want to change the actual species, as we want to go through succession, we've got to be shifting that ratio of fungi to bacteria. And so food graph, we looked at, I think we're up to 300,000 soil samples that we have looked at from around the world. We have published data on most of those systems, or other people have published the data. If you want to find out, let's say, citrus in Florida, what is the food web for citrus in Florida? You first have to go into the scientific literature and go and find 5 or 6 papers on the species diversity and the biomass of bacteria in citrus in Florida. Then you got to go find that same type of publication on fungi, and then on the protozoa and on the nematodes. because you cannot publish a single paper that goes over all of those things. In the scientific literature, the page limit is typically 3 pages. So if it's going to take 20 pages to describe the bacteria and the seasonal cycle and what kinds and species of bacteria, well, we're already beyond the limit of a typical single publication. And now we're going to put fungi in there, and then protozoa, and then nematodes, and then microarthropods, and maybe we should put earthworms in there. There's no place to publish a paper like that in the scientific literature. We were lucky when we got the ecological monograph published, because that was 75 pages. So we went through all of those treatments and described, and then looked at the nitrogen cycling and what happened to the plants. It's really rare to get an ecological monograph like that published. So the data are in the literature on all of these ecosystems. When you want to put that information together, you come talk to me, because I've put it together. It exists in our database at Soil Food Web. First couple years we worked in Soil Food Web. We worked in all of these agricultural systems, and we have a pretty good understanding for what the ratio of fungi to bacteria are for all of these different kinds of systems. Lots of samples, we've worked in lots of places, but you know what? I don't know everything. A scientist will actually admit to not knowing everything. I've only been, wouldn't it be boring if you knew everything? What would you do? I would be so bored. So I am really not in any danger of becoming bored in my life. I have lots yet to do. So when we're trying to establish what is the right fungal to bacterial biomass ratio, what we typically do is go to a watershed, because we want to be on the same soil type, we want to be in the same climate, everything held the same, and start looking at the successional process. So when we're in old growth forest, in this watershed, well, it's the same soil, same parent material. It's same climate. Now let's look at what happens when our animals are gaining 2 pounds of weight gain per animal per day in a 90-day feeding study. Will you agree with me that this is a lot more productive system than this one, where the animals are only gaining 1 pound of weight gain per day in a 90-day feeding study? Same number of animals, way more productive system. What does the biology look like in that soil? Not as predictive. What happens? And then we go to an agricultural soil. Both of these are oats, but in this soil we gained 180 bushels of oats versus 100. Which one's more productive? So looking at plant production in these different kinds of systems. Now let's look at the biomass of bacteria and the biomass of fungi. More than 2 times more fungi than bacteria in this system. This is a fungal dominated system. Is it as healthy as this forest system could be? No, it was a pretty sick forest. There were some weedy species in there were some dying trees, so it could be healthier, but it is definitely on the fungal dominated side, and that's what we see most of the time in any forest system that we're working with. Now, as we go to an earlier successional stage, not as productive. We're looking at really good production of animal weight gain. It's still a fungal dominated system. But look at how much more bacteria we have there. We actually have more bacteria at this stage of succession than at the latest stage. Still fungal dominated, but not nearly as. Now look at what happened. Fungi and bacteria as we lost productivity in that system. Why is this grassland not able to maintain weight gain in the animals? The plants aren't getting the nutrition that they require. They're not regrowing as fast. What is it that explains why this is not as productive an ecosystem as this pasture or this grassland? What did we lose? We lost both. Both of these have fallen below 300 micrograms. And what we see over and over around the world is that you will not have a productive ecosystem until you get a balance of these organisms. Both must be above 300. So when we're looking at a forest system, our bacteria have to be at least 300, well, and then our fungi are going to be 1,200. Fungal bacterial ratio proper, but the lowest organism, the biomass that's lowest, has to be above 300. No matter where we look on the planet, it has to be at least 300 for it to be a productive system. This is not productive. You're not going to be able to really raise the animals you want to raise if you're only gaining one pound weight per day. Now let's go into the agriculture field. Oats. Notice they're both above 300. It is slightly bacterial dominated. That's quite typical. Almost equally balanced. Fungi to bacteria when we're dealing with row crops. Barley, oats, corn, things like that. As we lose productivity, we've lost both sets of organisms, but what's really suffering is that fungal biomass. We are not productive enough typically to be able to keep the farm if we're only making 100 bushels of oats. Is this the chemical system or is that the chemical system? Which one's organic, which one's chemical? Yeah, that's the conventional, this is the organic. And they're doing that organic right. Now, what do we mean when we talk about this plate count index? So these are plate counts. When we first started out, there was a lot of, oh well, it's easier and cheaper to do plate counts. So we had people in all of these systems send samples into a plate count lab. And then they're looking at the number of species that grew on those plates times the number of individuals of each of those species occurring on those plates. They used 5 different media in order to grow 5 different types of groups of bacteria. And in the whole world forest system, notice that they didn't have anything grow on any of the plates. They had 6 species, 5 species here, they had 6 species there, oh whoopee. 7, 12. The laboratory actually called these people up and said, so, you don't have anything growing on any of the media. Is this a desert soil? And they kind of snorted and said, how about if you go look at the sample that we sent you, because it's almost completely organic matter. Nice, rich, dark, old growth forest soil. How come none of the bacteria in that old growth forest soil grew on any of those petri dishes?
No pathogens.
They don't grow in anaerobic conditions. No, so we didn't have any pathogens in that soil. None to speak of. So now look at, can you really differentiate those two systems using that plate count index? Yeah, and yet, look at how different the productivity is between those systems. You can't tell when you're looking at plate count data. How about this information? Which is the one that's got more diseases and more problems? The highest count of species of any of the ecosystems, because what we're really measuring with plate counts are the disease-causing organisms. That is not the way people typically interpret plate count data. They usually say, well, the more species, the better. So this would be the most productive system. Excuse me. Because what are we really measuring with plate counts? Which is the sickest. Yeah, what's the sickest system? So are plate counts useful? Yeah, but interpret them correctly. If you got lots of bacteria or fungi growing on your plates, you might suspect that that's a really unhealthy system. You got a lot of disease. So we have fun and stuff like that. Is it this scary? I'm going to actually make you look at all of this data. I only have about 5,000 or 6,000 tables like this one. Well, we're trying to say what is the proper active bacterial biomass or total bacterial biomass. active fungi, total fungi. What diameter? So diameter in a fungus, the narrow ones, diseases, wider ones, good guys. Protozoa, flagellates, and eviciliates. Nematodes, are these good guys or are they bad guys? So we do this over and over and over again to try to establish where the balances need to be. Fungi, bacteria, protozoa, nematodes. And on this side we have listed our ecosystems based on the plant material being produced in an annual budget. So not very productive, weeds disease. Chemical garden where they're using a lot of pesticides and you've got fertilizers and herbicides. Chemical pasture, organic pasture. Clear cut, so it's the overall forest system. Once the trees have been cut down, about 15 years after the trees have been cut down. So not as much plant material being produced because the trees aren't very tall yet. Organic garden, organic potatoes, strawberries, young Douglas fir, old growth Douglas fir. So these are watersheds in the Pacific Northwest, here where I live in Oregon. As you're looking at active bacteria, look at the fact that it stays about the same, maybe a little bit low there, until we get to mid-succession. And then it levels off when we get to the highest levels, the latest successional systems. Total bacteria, well, it's a lot higher than active, but again, it's staying about the same, and then it starts to pick up higher and higher. It levels off midway through succession, and then levels off. Active fungi, staying about the same line here, and now all of a sudden really picks up. Fungi take off as we get into those perennial systems. Total fungi, again, really picks up. So you can start to look at that ratio of fungi to bacteria and give yourself a really good idea of where you are on that productivity gradient. Highly, right there on the edge of being really productive, quite productive. Strawberries, actually right on the dominant side. Diameter of fungi, only disease-causing fungi here. Now it's starting to get a little bit better. The 3 is left over from the fact that this used to be an old-growth forest, and those fungi are still hanging around. The most beneficial fungi are late in that succession system. Protozoa, lots of protozoa. Look at how bouncy the data are. It's like all over. Highest levels, midway through succession, where bacteria are highest. Oh, how amazing. Bacterial feeders would be highest where you've got the most bacteria. And then almost nothing when we get into the fungal dominating systems. We're looking at nematodes, root-feeding nematodes early on in these systems. Now it's starting to pick up some bacterial feeders. So as the root feeders drop out, the bacteria start picking up. Now we pick up some fungal feeders a little bit later, fungal and bacterial and predatory nematodes. So trying to understand these balances, looking at data like this. Now the last column we were trying to come up with a simple index to explain all of these things, because when you work with bureaucrats, they cannot deal with this much data. Don't try to make them understand this. They want a numerical score that ranks everything. And I gave up on that, that just doesn't work. Drive me crazy. So what we've seen, the summarization of all of this, is that as we go through succession, we're starting out, Mount St. Helens explodes. Instantaneously, all of those 4 systems are volatilized into the atmosphere. So all of that life, all of that organic matter instantly just poof, and then all of that ash, the sand, silt, clays, everything falls back down to the surface of the soil. It is sterile. The first thing that comes in are the photosynthetic bacteria. The cyanobacteria, they have the enzymes to cellulose phosphorus out of the rocks, calcium out of the rocks. They have the enzymes to fix nitrogen. So the cyanobacteria, photosynthetic organisms, are going to be the first things that come into an ecosystem. There is not a chance that there is any plant that can possibly grow in that system, and the only thing you've got in that soil are bacteria. As soon as bacteria are in, they are producing organic material, releasing that into the environment, and so now other bacteria come in and start to grow. As we build up these components, we get enough bacteria growing that the protozoa can establish, and now we get some protozoan populations. Now we're cycling nitrogen, phosphorus, sulfur, magnesium, calcium, all of those things, but not yet enough to allow plants to be able to grow. Fungi arrive, from the bacterial biomass ratio, we have 100 times more bacterial biomass than fungal. Very bacterial dominated, and typically you cannot get a plant to grow unless you use inorganic fertilizers. So yeah, you destroy your life, the only way you can get plants to grow in that system is to try to constantly feed them with the soluble inorganic materials. Nematodes arrive, microarthropods. Now we've got enough of a food web, enough nutrient cycling, that we can get weeds growing in our system. As the bacteria started to grow, the protozoa come along behind them and started to eat them. And as the protozoa start to eat the bacteria, they're releasing nitrogen in a plant available form. So that pulse of plant available nitrogen being released, the weeds go, look at this, we got massive amounts of nitrate, germinate, grow, suck the nitrate out of the soil, produce those seeds, and now as the protozoa are eating those bacteria so fast that the bacterial population starts to crash, so the protozoan starts to crash, and as now all the bacteria are sucking up whatever nutrients are available as the bacteria grow, because the protozoa are now down here at nobody home, bacteria start taking up all the nutrients. There is no plant, even weeds that can grow. Well, as those bacteria are taken off, the protozoa go, hey, wait a minute, there's lots of bacteria. I can start growing again. So you're going to have protozoa, big pulse of nitrate out into the soil. And then so here, there goes our weeds, taken off, growing really happily. But as the protozoa overeat your bacteria and everything crashes, our later successional plants can't tolerate that pulse of a huge amount of nitrogen, nothing. What? Weeds are designed for that kind of system, and they do very, very well. Weeds don't put much of their energy into root production. Weeds don't need roots, except to suck up that big pulse of nutrients that's being released by the bacterium protozoa. So weeds only put about 20% of their energy into the soil. The rest of it goes to producing seeds. Next generation disperses and they repeat that system. So they don't care whether your soil is compacted. You're not putting their roots down far enough to worry about it. So these early successional systems are going to be selected even for the growth of weeds. The bacterial biomass ratio, 10 times more bacteria than fungi. But weeds are producing plant material. We're starting to get some different kinds of foods being produced. We do have some cellulose, we've got some lignin. So there's a little tiny bit of fungal food going down under the surface of the soil when the weeds die. And at least there's some roots going into that soil, it's better than nothing. Everything on this planet has been through the weed stage of life. Because you can't get from here to there without allowing the weeds to start this whole process. More fungal foods than bacteria. And so with enough of this weed plants growing, we are eventually going to shift and change that ratio from fungi to bacteria. And now we set the stage to grow our early successional grasses, our Bermuda grasses, our Bromus. The wetlands, the riparian plants go in here as well, typically. Broccoli, cauliflower, the brassica, the mustards, non-mycorrhizal plants in this category. And at least you got to have more fungi. They don't do well here. bacterial. So eventually, with time, we're going to shift that ratio, and now other grasses, our veggies, can start growing. Ratio getting close to equal biomass. 0.75, 0.8, somewhere around in there. They're putting in more fungal foods than bacterial foods. And so we're going to start shifting that biology in that soil. So eventually, 1-to-1 ratio fungi to bacteria. These guys are no longer being selected for. Now we've got our late successional grasses, our row crops, our bents, our fescues, highly productive grasses, corn, wheat, barley, those sorts of things. Equal biomass. And these plants, of course, put way more fungal foods into the soil. So we're going to start shifting pretty rapidly into a truly fungal dominated system. 2 times more fungi than bacteria, but 5 times more fungi than bacteria. And we're going to be growing shrubs. We're going to be growing vines, bushes. And they, of course, are making woody materials. A lot more fungal. So we shift into early successional deciduous trees, later successional, lots more fungal foods going in the system, and we're going to set the stage to move into the old world species. Takes a while, doesn't it? So Mother Nature uses that balance of fungi and bacteria, and then of course the protozoa and the nematodes follow right along. What prey is available? So what predator is going to be predominant? Fungal feeding predators are predominant here. Bacterial feeding predators are predominant there. So Mother Nature pushes succession by ever increasing the fungal dominance within our soil. And we have lots and lots of data to show that that is what's pushing that system. Question?
So, without doing our microscope testing, which we want to do, but if we've seen just, if we have multiple crops growing together and the mustard goes ballistic and everything else is kind of medium growth or bacterial dominant.
Probably, yeah. So look at what's actually growing in your soil, and you're going to get a pretty good idea of what the biology is in there.
I'm sorry, but this is a little off. For your aquaponics people that you've taken biological, or your hydroponics people that you've taken biological, can you establish fungal colonies in your water colony?
Yep. Because what we really have to do is get these sets of organisms established on the root systems. And so, of course, the plant is putting out the exudates to grow bacteria, grow fungi. Because now, remember, the plant here is not just kind of a passive, stuff, whatever's out there. Those plants are going to be putting out exudates to try to maintain the system. They really don't want to have things shift and go to the next stage. Of course, their dead plant material is going to push it that direction eventually, but it can take a long time. So those plants are trying to maintain the proper ratio for them. And they're pretty effective in aquatic systems. We can maintain that fungal to bacterial biomass ratio around the root systems of our plants in those hydroponic systems. We don't have to be putting in soluble inorganic forms of those nutrients. And your plant is really good at knowing exactly what it wants.
I have heard basically that a lot of times where the trees grow, it's like people think nothing else really will grow there. Like it won't support vegetable growth, but it'll support trees to grow. And so that because the soil is poorer, like, and this seems to be kind of the opposite.
It's poorer for the growth of all of these guys. Not because we lack nutrients, it's because we're self-dominated. So does it really make that soil poorer? It makes it a different stage of succession.
Looking at like some of these maps where they show from one side of the country to the other, like how much the soil has had water eroding on the trees and such, and then where the soil is not as eroded, and that's where it doesn't really have as much like available nutrients or something, and so that's where the trees will grow, or it's it's been so wet for so long that only trees will want to grow there or something. I mean,
There's not a lot of rhyme or reason when you try to really understand what they're suggesting. It's hard to figure out why does this swampy area doesn't have trees and then why this swamp area does. You really have to start looking at that ratio of fungi to bacteria in the soil. This is very bacterial. This has massive amounts of fungi. How did we get the massive amounts of fungi in that cedar swamp? But you gotta look at past. You look at this swamp and why is it strictly bacterial? Because it's anaerobic as all get out and we've lost all the fungi. So starting to understand that this successional process is actually driven by the biology in the soil. And people trying to decide that this is poor soil because trees are growing here. It's not poor. Cut the trees down, change the biology in that soil, and we can grow some really good corn or wheat or veggies or whatever you want to. So where do those nutrients suddenly come from? By the biology. Yeah, it's not poor soil. It's not poor soil at all. You just have the wrong ratio of fungi to bacteria to grow corn. Yeah. So if you were going to go into one of these forest systems, what's the easiest way to convert this forest into a system where you're going to grow corn? Wow. Love it. Till it up, slice and dice the fungi. So Europeans coming to the east coast of the United States, created by those beautiful chestnut forests. What did we do to it? Yeah, we cut it down, ripped the root systems out, plowed the living daylights out of it, and we grew great corn for a while. Yeah. Or we grew tobacco in it. Now there's a really stupid idea. So, can you deplete the biology in the soil? Can you destroy? Yeah, absolutely, and that's what we've done. So, another way of thinking about this, maybe a little bit easier for most people, is to actually put values on that bacterial biomass and that fungal biomass. Early in succession, no plants. A little bit later, we have a few fungi in the system, but we're set in stage to grow weeds. Now we're in that mid-successional veggies. Now that mid-successional, really productive grasslands. Moving on to perennial shrubs and trees, and old growth shrubs and trees. So maybe a little bit easier for you to understand it in this fashion. Now, there's a whole other bit of information that we've got to go into, is to understand how this shift from fungi to bacteria affects chemistry in the soil. So we're going to be going down that pathway tomorrow. So when we're in that early successional system, bacterial dominated, almost the only form of nitrogen is going to be nitrate. As we start to pick up the fungi, as fungi start to become more important, now we start to produce ammonium. So strictly bacterial early in succession, strictly nitrate. Start to add some fungi, now we go. Think about it, we start off with lots of nitrate. And then we go to the next stage of succession. Now we've got some ammonium, and that nitrate is not as predominant. Go to the next stage of succession because we've got more fungi, we're going to get more ammonium in that soil, less nitrate. Now we're growing our most productive grasslands, row crops, equal biomass of nitrate and ammonium. Your corn has to have ammonium, or it's not going to be doing well at all. As we go over the edge into perennial systems, now more ammonium than nitrate, because more fungi than bacteria. We go into our deciduous. Here we go, exactly the other way around, to the point where we're in a truly healthy old-growth forest. You will not find any nitrate to speak of. It's all ammonium. And that ratio is determined by the ratio of fungi to bacteria. Now, why does it work that way? Okay, so we're going to have to go into an understanding of the chemistry of the system, and I'm going to try to make it not too difficult to follow me through that one. So whenever I start using it, stop me. wait, I don't understand how we got here. So I'll try to go back for it. So any questions about succession? How come everything on this planet is not an old growth forest? Disturbance. Because of disturbance. So take a look at this landscape. Back when Europeans arrived in this part of the world, this was all old growth forest. Europeans came and mowed it all down. Now, because they left it alone, look at the mature forest that has regrown in this part of that landscape. How come this has not recovered and become a mature forest? How come this hasn't either?
It's great.
Yeah, because of that disturbance. This is too many sheep not being controlled properly. And the sheep spend most of their time in the waterways, and so they are causing this erosion. We don't have the proper plants coming back. They're grazing down the trees, so this ecosystem constantly disturbed. We have a lot of weeds, a lot of problem organisms here. Now, exactly the same number of sheep over there as over here, but they are so great. Look at the fact that over here we don't have the erosion, we don't have the damage. Because those animals are grazing intensively for a short period of time, but they're moved on before they can completely denude that soil of plants that are in the system. Because of the disturbance, these areas have not come back to the mature forest. That would be possible had that disturbance not been imposed on this land. So, how come we've got this really nice sharp line of demarcation between here and there? That's not a good man. So, disturbance, critically important to understand that. So, lots of different kinds of disturbances: compaction, fertilizers, pesticides, wiping out that biology, moving us backwards in succession. Unusual weather events. It freezes in July. We have 80-degree weather in December, and then it freezes. So all of these kinds of disturbances can be moving us backwards along that successional scheme. A flood may not actually move us backwards in succession if the flood occurs in the cold period of the year. But if a flood occurs when the microorganisms are growing rapidly in the soil, and they drive that soil anaerobic, well, you're going backwards quite some distance. Insect plagues, so how many insects? It's the intensity and frequency of the disturbance that determines how badly we're going to go backwards. Think about Yellowstone National Park. It's got to be about 30 years ago, we had a devastating fire, burned a great deal of the park. So there were actually Congress people that said, well, we should sell Yellowstone because it's been destroyed, sell it to people because, we could use the money. Right? Like succession doesn't happen. Now, some parts of the park, the fire went through just as a really quick understory burn. Didn't kill the trees. The next year, the plants did, the trees just grew a little bit faster, larger growth rate. Some parts of the park actually burned the trees, lost the trees, and the next year, some beautiful pasture, better grazing for the animals. But because the cones are serotinous cones that would germinate after fire, the system started to regrow the trees. We're already back to those trees are 3 or 4 times taller than I am. Some areas, it actually started to burn the organic matter in the soil. And so we went back to weedy systems. Most of those weedy areas are now proceeding through succession. Some of them are already proceeded down to here. Some of them are still stuck there. We don't know why. Some of this is, I can't explain everything sometimes. The fungal-to-bacterial biomass ratio is not shifting. Some places, Wayne, it's already down here. So why? Can't always explain that. Some places in that old growth forest, there was so much fuel load on the soil surface that it got the temperature hot enough, long enough, that it caused the silica in the soil to melt. and the surface of the soil turned into glass. Now, they invite me back to Yellowstone every once in a while to take a look at the biology going on in those soils. And 2 years ago, I was there to take a look at these areas. So 28 years or something like that had passed before the last of those areas started to grow anything. Some started to recover right away. Some of them, it has taken 30 years basically for them to start to recover, but they are recovering. How long will it take for them to become old growth forest again? As long as we don't disturb it, as long as nature doesn't disturb it. It could get there pretty fast, or it could take 1,000 years. It depends on all these other disturbance factors. So, how important is understanding disturbance in your system?
How is it that in our fresh lava, the first thing that grows is not a tree?
It's not the first thing that grows.
It's like I mean, if we go out there, I mean, you can see it. It's like.
First thing that I've observed is lichen.
And I think it's that you have to have an accumulation of organic matter in there. So what I've observed is that the place the trees or the palm trees start growing is where you have organic matter from outside the system flow into the cracks and crevices. And then that's what allows those trees to germinate. But you gotta have enough highly fungal material flow in there to allow those seeds, allow the coconuts to actually germinate and start to grow. So lichens coming in first, starting to give us a little bit of surface structure, so we can hold on to that organic matter, and then we can kind of jump start. So do we always have to go through? Around those trees, we're not going to be up here right away. The rest of the system has to come along too. So I bet we have some other interesting lessons to learn too when we get into some of these different systems. We don't know everything. So if we look at this in the box and arrow, it's kind of cute. So this is that same food web, but now it's in boxes. The organic matter coming from shoots and roots. Dead plant material from other organism biomass from all the other boxes. Fungi or bacteria, simple or complex. Mycorrhizal fungi getting their sugar straight from the plant. Nematodes, we need phytophthora nematodes. And when we're looking at soils that are growing lawns or vegetables or row crops, the fungi and the bacteria generally are fairly well balanced. We have more or less an equal number of bacterial feeders and fungal feeders. And if you have your biology like this, you're setting the stage to grow these intermediate sets of plants. What if we want to grow a forest? Well, we've got to improve, we've got to increase that fungal component. And so we're going to increase the fungal predators. So healthy orchards, healthy forests, much more reliance on these fungal forms within the system, get the chemistry right, let the biology and the soil control the chemistry that your plant requires. And if you leave your soil alone and nothing else but weeds grow, then you can bet that this is what your food web looks like. So can you use the plants to tell you what is probably in your soil? Yes. So start looking at the plants and get an idea of where you are in succession. Does biology affect soil chemistry? Well, we talked a little bit about soluble nutrients, so hopefully you can understand soluble nutrients are all salts. All soluble nutrients are salts. That means all organic, inorganic fertilizers are salts. Now most people get a little confused by that. The only kind of salt they've ever known about is sodium chloride, table salt. But there are hundreds and thousands of other kinds of salts. Anything that dissolves in water is going to be a salt. Any inorganic mineral material is going to be a salt. So when you start looking at calcium carbonate, is that a salt? No. There's the positive part of the molecule, there's the negative. And so when you put that into water, this material dissolves because it's a salt. The hydrogen complexes with the carboxide, the calcium complexes with the negative part of your water molecule, and it dissolves. It disappears in that water. Well, salt is the same thing, table salt. We're going to have our hydroxide binds with the positive part of that molecule. Your hydrogen, positive, binds with the negative part of your salt molecule, and it dissolves in water. All inorganic fertilizers are salts, and you are taking water away from your plants. But long before you ever affect your plant, you are going to be killing the biology in your soil. So bacteria, fungi, protozoa, nematodes, much more sensitive to any of the salts. So think of the common fertilizers. Ammonium nitrate. Positive, negative. You put that into your soil, you're taking water away. You're killing things if you're putting more than just a little bit of that inorganic fertilizer into your soil. We're killing stuff. So, when you put an inorganic fertilizer into your soil, what are you doing to the biology? We're losing it. We're starting down that pathway of chemical dependency. Why don't we call it an addiction? Because that's what it is. We're killing the things that allow us not to have to use them. So, mineral, typically not organic, typically forms of salt, so just some definitions. When we're talking about organic, what do we really mean when we say organic? A lot of people will say organic just means it's got carbon in it. So, carbon dioxide, is that organic? It's got carbon in it. It's not organic. This is the mineral form of carbon. So if we're looking at CH4, methane, is that organic? No. When we're looking at calcium carbonate, is that organic? Nope. It's got carbon in it. So what's the actual definition of organic? We mean a carbon chain. So we've got to have a chain of carbon, we've got to have photosynthesis. Those carbons are linked together through sunlight energy. That's what makes it organic. Mineralized, this is a problem. Soils people especially are really bad at misusing these two terms. Mineralized and immobilized. When we mineralize something, we're converting it from organic to mineral, typically salt. When we immobilize something, that means we're converting it into an organic form. Our nutrients are immobilized in the biology. They're immobilized in the organic material. We mineralize them when we take that organic form and spit out the salt. Why is all this important? Because guess what? We're going to do the nitrogen cycle. We're going to understand this whole process. So, we've been through this already, so I can zip through it pretty quick. Sunlight, CO2, produce sugars. There we go. We're on our way. What does your plant do with that sugar?
Bribes the little guy in the soil to bring stuff.
So yeah, that's a good definition. So this sugar is going to have to go down into that root system. There it goes. Now we got the sugar in the root system. Great. What does your plant do with that sugar? Well, what does that plant need to get from the root system? So we're going to have to pull the nitrogen inside. We're going to have to pull the phosphorus, the sulfur, magnesium, calcium into the root system. And we have to bind those nutrients on our sugars. So let's go through nutrient uptake in the soil. Kind of keep that one in mind because I've kind of given you the answer to some of the things that I'm going to be asking you about in just a minute. Typical explanation of nutrient uptake when you're dealing with a soil chemist or even plant physiologist. The way plants, the there's our root, the way the plant takes up nitrogen or phosphorus or salt, or magnesium, is it has to be in the inorganic soluble form. And then through simple diffusion, this molecule, these molecules are going to diffuse into the root system. And so we're going to have the same concentration of nitrate outside the root as we have inside the root. So let's draw that. Here we go. Same concentration inside and outside, so our plant is taking up nitrogen. Except nitrate is not usable by your plant. That can't be wandering around through the phloem and the xylem of your plant because it's going to kill your plant. So what is the form of nitrogen that your plant uses to wander around inside the body of the plant? Because it can't stay in this form. It's unusable to the plant. How do you link nitrate up to that sugar molecule, which is what's going to escort your mineral nutrients around through your plant so your plant can actually grow? How do we attach this to that sugar molecule? And that's really what the sugar is doing, going down into the root system. If we want to think about the fact that if we equalize concentration of this nitrate inside and outside the root, then that's it. Your plant's not taking up any more nitrogen. End of story, your plant's going to die. So we have to convert that nitrate into a different form so that diffusion will keep pulling that nitrate or ammonium into the root. Because if it's just kept as that simple, well, through simple diffusion, it moves into the root. Well, then so what? You're not going to make the nutrient that the plant requires. So, if we exit those 3 oxygens, blow them off, the plant doesn't need oxygen. Not in a nutrient form. So one of that nitrogen is going to get bound to that carbon. And along the way, we got to pull in 2 hydrogens. So we're taking 2 hydrogens out of our soil, binding it to our nitrogen, hooking it onto our carbon chain, and now this is an amino acid. We're going to make a protein. What's going to happen to our soil pH if we're pulling in 2 hydrogens? It's going more out. So when we take out nitrate and we hook it to our carbon chain, so this can now take off and be used to grow our plant, proteins, make enzymes, make DNA, make all of those wonderful things, we're going to actually cause the soil around that root system to become more alkaline. So why do they always say when plants take up nitrogen, it causes the soil around the plant to become acidic? Does it make sense, doesn't it? Can't be happening when we're doing this. So there's a common mistake that they're doing in most plant studies. They pay no attention to what's going on with biology out here. Only in the instance where we have anaerobic conditions occurring in our plant study will this become more acidic. Isn't that fine? So because they're seeing nitrogen uptake, and they're seeing acidic conditions occurring in the soil, they're assuming, when you assume something, it makes an ass out of you and me. They assume the 2 things are correlated, and they're not. So, we've got to go beyond the conventional explanation of how plants take up nutrients. Plants take up nutrients and they have to convert them. And that conversion is going to change the pH in our soil typically. And we've got to understand that conversion, that's what's actually happening when our plants take up nutrients. Except, what I just told you is not true. I just lied to you. Didn't it sound good? What's actually going on here? Because there's something else you got to think about. What about the soil biology around the root system? Yeah, so around that root, we've got billions of bacteria growing. we got miles of fungal hyphae being produced. And now the fact is that when that nitrate starts diffusing towards your root system, it's going to be intercepted by the bacteria and fungi long before it gets into your root. So all your insoluble inorganic forms of any nutrient are going to be grabbed by those bacteria and fungi. So how can your plant grow? Because there is no plant on the planet that doesn't grow bacteria and fungi around its root system or around its leaves. So we're going to have to understand this just a little bit. There's got to be more to the story. So what's the mechanisms? Well,
even these plants that are being fed the inorganic fertilizers, they have that.
yep. So, well, if you're trying to grow your plants in sterile media, yeah, okay, you can try to pump these things into the root system, but we don't do a very good job. The plant doesn't taste very good, and the nutrients are not balanced, so there's a lot of problems. Can we do it? Yeah, but they're not really good plants. So, gotta go back and, we're gonna have to understand that whole system because there's clearly a mental work going on here. So, well, how about if we start on this section tomorrow?
Yeah.
Go ahead and end just a little bit early because this next part really takes some concentration. So go get well rested. Go make sure you're ready for the big day tomorrow where we put it all together. See if you can figure out what's actually going on. When we have those bacteria and fungi on their root system, how do we make those things the opposite of reality?
Okay, you guys, can we start on time tomorrow? At 9? Okay, thank you very much, everybody.